Hello guys, welcome to today's video. Today I shall be taking you through structure of DNA or DNA structure. DNA, as you all know, is the hereditary unit of life in all living cells. DNA is the basis for which traits are passed from one generation to another in organism. Today we want to look at the structure of DNA. I believe that most of you are familiar seeing a shape like this by the chromosome, the helix structure. But we want to zoom in and take a closer look at the structure of DNA beyond just this arrangement. Now, DNA is made up of repeating units of a building block called nucleotide. Repeating unit of a building block called nucleotide. So we shall be taking a closer look at the structure of nucleotide. And once we understand that, we can see how it is aligned one after the other to give us the bigger picture of the DNA. So the nucleotide is made up of three components. The first component of the nucleotide is what we may call the sugar molecule, or simply put, the pentose sugar molecule. Now, there are two kinds of pentose sugar molecule. We have the ribose sugar and the deoxyribose sugar. So DNA derives its name from deoxynucleic acids. Whereas we have closely related to it, we have its sister called the RNA, which is the ribonucleic acid. This is the deoxynucleic acid. So two kinds of nucleic acids, deoxynucleic acids and ribonucleic acids. In one of my subsequent videos, I'll be taking a closer look at the differences between RNA and DNA. But for now, we are interested in the structure of the DNA and then, as by implication, that of the RNA as well. So it consists of sugar molecule. The sugar molecule, I just said, is a pentose sugar. That's because it is five-sided. 1, 2, 3, 4, 5. Pent from 5, pentagon. Now we also have 6-sided hexagon. We have 7-sided, 8-sided, and so on. But in this case, the number one component of the nucleotide is the pentose sugar. Now, in addition to the pentose sugar, we have another component, which I'll just indicate by the letter B, the nucleobase. So we have the nucleobase, we have the pentose sugar, and then we have the phosphate group. These are the three components of the nucleotide. What we have here is the structure of the nucleotide. This is the nucleotide. The nucleotide consisting of three components: base, sugar, and phosphate group. The sugar, I just said, is a pentose sugar. So this pentose sugar has 5 carbon atoms. This is carbon 1. By the way, here is an oxygen. This is carbon 1. This is carbon 2. This is carbon 3. This is carbon 4. This is carbon 5. Take note where carbon 5 is. So you don't start naming here. There's no carbon here. This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. And then this is the phosphate group. Now, each of these is attached. There's a hydroxy group here, but I don't want to draw all of that, so I wouldn't get it dirty or messed up. So there's a hydroxy group here. And here there is also a hydrogen here and a hydroxy group here. This is a hydrogen group, I beg your pardon, a hydroxy group here. In the case of an RNA, if it's an RNA, here will be hydroxy. But if it's a DNA, here will just be hydrogen. This is why DNA is called deoxy. Strictly speaking, we could have called it 2-deoxy. 2-deoxy, ribonucleic acid, nucleic acids, okay? Because 2 indicating the carbon number where there is the deoxy. You know, deoxy just means like without the hydroxy group or without oxygen. Without oxygen. Just like you have emphasize, de-emphasize, escalate, de-escalate. If one tells you to de-escalate, that means you should escalate, or you should go back, you should remove the escalation. Okay. If I tell you to de-emphasize, it means I'm telling you to unemphasize. All right. So this deoxy means without oxygen. So this absence of this oxygen is strictly speaking on carbon number 2. So that's why we can as well say, according to IUPAC nomenclature, we could write that name properly as 2-deoxyribonucleic acid. So this is carbon 3. It has its own hydroxy group present, no challenge. So the hydroxy group here is still here. Here we have the hydrogen, and here we have the carbon 5. The carbon 5 has its own hydrogen and then attached to phosphate group. The phosphate group could be 1, in which case you have nucleoside monophosphate. The phosphate group could be 2. In that case, you have nucleoside diphosphate. There could be 3. In that case, you have nucleoside triphosphate. But we're keeping it simple, so we just concentrate on one phosphate group for now. All right. So now, this structure we have here that I said is made up of base, sugar, and phosphate group. The sugar being a ribose sugar if it is an RNA, or deoxyribose sugar if it is a DNA. So in this case, what we have here is a deoxyribose, because you can see there is absence of oxygen on carbon 2. So this is 2-deoxyribose nucleic acids. Please take note of that. Now, the DNA can be said to, so the whole of this unit now is called a nucleotide. Nucleotide. This unit that consists of the base is called the nucleobase. So this is nucleobase. Now there are 4 kinds of nucleobases. They vary, 4 bases. We have the adenine, we have the cytosine, we have the thymine, and we have the guanine. This is in the case of DNA. However, if it is an RNA, if this nucleotide is for an RNA, instead of your thymine, you're going to have a uracil. So all the time in RNA, thymine is replaced by the base uracil. But generally speaking, adenine, cytosine, and guanine, they remain intact both in DNA and in RNA. So these are the 4 bases. These 4 bases, they vary. They vary from nucleotide to nucleotide. They are not fixed. But this component of the nucleotide, which is the sugar component, and the phosphate component, these ones are fixed. They do not change. Hence, they are called the backbone of the DNA. So this and this consist the fixed, or the backbone. We can call them the backbone, of the DNA. Now, the base dimension alone is called the nucleobase. But when you add the base and the sugar, then you have what we call the nucleoside. So this is nucleoside. Are we together? So base plus sugar is called nucleoside. Base alone is called nucleobase. The base and sugar is called nucleoside. Then base, sugar, and phosphate group are called nucleotide. So this is one unit of the building block of DNA. So a unit of the building block of DNA is called the nucleotide. So you're going to have repetition of this nucleotide, that gives you the complete structure of the DNA. So for simplicity's sake, let us draw another nucleotide here. It's carbon 5. This is base. And sorry, this is base. And then this is phosphate group. This is your sugar. So you have carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So the nucleotide has two poles, has two ends at any given time. You have a free end here, which is at carbon 3, and then we have a free end here, which is at carbon 5. So this place can be called 5' end, and this end here can be called 3' end. So normally speaking, generally speaking, this is the way the structure looks. There's now going to be another sugar here, another nucleotide here, and a base here, and something like this, and a phosphate group here. And then this carbon 5 is going to link to this carbon, the next nucleobase, via carbon 3. Similarly, you have another building block again, this way, a base, carbon 5, phosphate group. Phosphate carbon 5 here gets linked to carbon 3 via this bond. Then you're going to have yet another building block this way, a base, again carbon 5, phosphate group this way, and then carbon 5 of this nucleotide gets connected or bonded to the next nucleotide via carbon 3 at this end. So this is then there will now be a free carbon 3 end here. So this is like 3' end, and this is like 5' end. So at any point that you cut this DNA, the strand of the DNA, you will always have a 3' end that will be free and a 5' end that will also be free. So generally speaking, you got a base, carbon, sugar. So this is like the skeleton. This is like the backbone. So the backbone is made up of sugar, okay, carbon 5. Sorry, I beg your pardon. This is linking through the base. Beg your pardon. Sorry about that. Linking via the base. So sorry about that. My structure was a bit misleading. Yeah, something like that. That's better. My apologies. So the carbon 3 here is connected to the phosphate group. The carbon 3 of this nucleotide is linked to the phosphate group. The carbon 3 of this is linked to the phosphate group. In other words, is linked to carbon 5 via the phosphate group. So this bond here is called the phosphodiester bond. Okay? The phosphodiester bond. So this is the bond that connects carbon 3 of one nucleotide to carbon 5 of the next nucleotide via the phosphate group. Okay, so this is sugar. What you got looks like the sugar, carbon 5, phosphate group, carbon 3, sugar, carbon 5, phosphate group, carbon 3, sugar. So just like that, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then you could just look at it like sugar, phosphate, phosphodiester bond, sugar again, phosphodiester bond, sugar again, phosphate bond, sugar, just like that. So this is like this, the backbone. This backbone is fixed. It doesn't change. It's just that way, stagnant. However, the angle, the other side here, that constitute the nucleobases, that side is variable. So it could vary to the degree of 4. It could either be a cytosine that could be here, either a cytosine or a guanine or an adenine or yet another adenine. So this could be the basis that could be varying here. This region here, it varies. It varies from one nucleobase to another, from one nucleobase to another. That is the way this varies. But however, this angle here, the sugar and the phosphate group, they remain fixed. Sugar and phosphate group, they remain fixed. So the nucleobases are the ones that are varying. And the variation of the nucleobases is actually what brings about uniqueness from one individual to another. It is this variation also that determines the nature of the gene or the nature of the hereditary material. It is this variation that tells you the information that is carried in the hereditary unit. The information is being determined here. So if you're going to sequence this strand of DNA, you may have the sequence to read something like A, G, C, okay? So this side is variable, but this side is unvariable. So by and large, you're going to also have the next strand, which will be like upside down or anti-parallel to this one. It will go something like this. Pardon, let's take it up a bit. So you're going to have this going down, this going down. This will be the complementary strand. Complementary strand might be going down this way. So take note, you have your oxygen here and your oxygen here. Your carbon 5 will be shooting out here. There will be a phosphate group here. This phosphate, sorry, beg your pardon. Carbon will be shooting down here. There will be a phosphate group here. Yes, this way, and connecting to this like this. This will go like this. Phosphate group here, and we connect to this is carbon 5 phosphate group here, which will connect to the next one like this. Okay, so in this case, you have here carbon 1. This is your carbon 1, carbon 2, this is your carbon 3, this is your carbon 4, your carbon 5. Okay, whereas here you have your carbon 1, carbon 2, carbon 3, carbon 4. Take note that the bases are always attached to carbon 1. The bases are always attached to carbon 1. So if you take a good look at this, you're going to see that your carbon 1, sorry, your carbon 1 carries the base. So there's going to be another base here that will be complement to this. Since here is an adenine base, here will surely be a thymine base. And since here is an adenine, here will also be a thymine. Since here is a guanine, here will be a cytosine. Since here is a cytosine, here will be a guanine. So adenine we always pair with thymine by double hydrogen bond, while guanine we always pair with cytosine by a triple hydrogen bond. So the bond that holds this strand and the other strand together, the inter-strand bonds, they are hydrogen bonds. So between adenine and thymine, you have double of it. That is double hydrogen bonds. That's the strength of the bond. And then between another adenine and thymine, you have double hydrogen bonds. But between guanine and cytosine GC bonds, they are triple hydrogen bonds. And then this is yet another GC bond, triple hydrogen bonds. All right. So take note that adenine always bonds with thymine complementary, while guanine is bonds complementarily to cytosine. So these strands here, the end of this, the free end of this strand here is 3', whereas the free end over here is 5'. Take a look at that. It is opposite to this. So we say DNA is anti-parallel, all right? Because one strand is ending or starting in a 3' here and ending in 5', while the other one is just starting in a 3' here and ending in a 5'. It is anti-parallel. Okay, so DNA strands are complementary to each other, complementary in the sense that adenine will bond with thymine, guanine will bond with cytosine. If here is cytosine, here will be guanine. If over here is thymine, here will be adenine. If it is adenine, here will be thymine, and just that way. So the bonds that hold the two strands together, they are called hydrogen bonds. This is hydrogen bonds. If it is between adenine and thymine, it is double hydrogen bond. If it is between guanine and cytosine, it is triple hydrogen bond. If it is between cytosine and guanine, it is still 3 hydrogen bonds. So there are basically 3 kinds of bonds here that we might identify. First of all, I mentioned the phosphodiester bond, which bonds carbon 5 of one nucleotide to carbon 3 of the subsequent or the next nucleotide. This bond, I said, we call it phosphodiester bond. Second bond we just identified now is the bond that holds the 2 strands together. That means the bonds that holds the base, nucleobase of one strand or one nucleotide with a corresponding or adjacent corresponding nucleobase from the other strand. This bond is called hydrogen bond. Then there's a third kind of bond that I'm yet to identify here. That is the bond that holds the nucleobase and the sugar molecule. There's a bond that holds this nucleobase, this cytosine, and the sugar molecule. I'm talking about this bond here. So this bond is called the glycosidic bond. Glycosidic bond. So these are the 3 bonds that you have. So take note why this structure is just opposite. Let's say why this structure is going down, this structure is facing up. So they are flowing, they are moving in different directions, okay? One goes up like this, the other is going down this way. So there are 2, and there are bonds. There are bonds that hold them from one strand to another. There is a bond, hydrogen bond holds them together, while a phosphodiester bond holds one nucleotide to the adjacent nucleotide within the same strand. Whereas glycosidic bonds holds the sugar molecule of one nucleotide to its corresponding nucleobase. So there you have it, the structure of nucleotides, or by and large, the structure of DNA. So DNA is just a repetition. It's a macromolecule. It's a big molecule. It's a polymer of nucleotide. It's a repetition of this nucleotide. But if this was to be an RNA, instead of a T here, you are going to have a U, a uracil. Instead of this T here, you have a uracil. All right. Then if it's an RNA, instead of having a deoxy here or the missing oxygen here, you are going to have a hydroxyl group here. There will be oxygen here. And when there's oxygen here, that will make OH, and that will be a hydroxyl group. So in that case, that will be ribonucleic acid, meaning that the sugar becomes a ribose sugar. The sugar is now a ribose because it has a hydroxyl group. But if there is no hydroxyl group, it's just a hydrogen group, the sugar is missing an oxygen, and then you call that kind of sugar deoxyribonucleic acids. All right. So I hope that with this explanation, you have come up to speed with your understanding of the basic structure of DNA. So it is these strands that we have here that are arranged and twisted in this form to give you this structure you have always known as the double helix. So this is the structure we have. We have bonds holding one strand and the other strand, these hydrogen bonds. They are holding one strand to another. There are hydrogen bonds holding one strand to another in a complementary way. In other words, if there's an A here, there's a T here. If there's a G here, there's a C here. And then there are also strands, bonds that are holding one nucleotide to the next nucleotide. Those bonds, we call them phosphodiester bonds. They hold one nucleotide to the next nucleotide. There is a bond that holds the adjacent nucleotides within the same strands together. Phosphodiester bonds holds 2 nucleotides within the same strand together via the phosphate. It could be 1 phosphate group or 2 phosphate group. It could be monophosphate, diphosphate, or triphosphate. All right. So I think with that you have some fairly good understanding of the structure of DNA. Don't forget to like the video and share and subscribe. I'll see you in my subsequent video.